I'm here to present my presentation on systemic acquired resistance chemicals, which we are using against the foliar plant diseases in onion and roses. And just a background of that, uh, plants we know, they are continuously facing various
You have to
challenges.
unmute. We're not hearing you.
Hello, yes?
No, we're not hearing. I'm not hearing.
Not here. So we know that plants, they are continuously facing various this biotic and abiotic stresses. And as a response to these stresses, they are activating some of the several morphological, biochemical, molecular mechanisms, which are having some signaling pathways. And as a plant, they have developed advanced immune system against these biotic stresses. And first line of defense, that is a passive mode of action, and thereafter the chemical compounds they get triggered from the signals received from the pathogens in the plant system. Two levels of the pathogen recognition that is there. We call it as a PAM, that pathogen associated molecule patterns are there. And second level of the immune system, which the plant respond, is based on the resistance genes, or proteins, which they recognize as AVR proteins, because they can trigger the effector triggered immunity thereafter. And hypersensitive responses, as well as the programmed cell death, is the response which is going on in the plant system, and they are warding the pathogens against their infection. So most of the R genes, they are coding for specific domain, and thereafter we have seen that some of the activations in the system of the membrane of the cell wall that takes place, and different ion channels, they get activated, including the elevation of the cytoplasm calcium level. Thereafter, some of the ROS chemicals like reactive oxygen species and activation of the mitogen activated protein kinases, they get activated. And similarly, in downstream signaling pathways, we have seen that certain hormones, which are called as the salicylic acid, they are active against the biotrophic and the hemibiotropic pathogens. And in turn, they activate the whole system, not localized, but the whole of the plant system. That's why we call it as a systemic resistant responses. And they promote the expression of the PR genes that provides a long-term defense against the pathogens. And exogenous chemicals can also be induced into the plants to increase those reactions. Similarly, we have seen that jasmonic acid and ethylene, these pathways get activated against non-pathogenic or biological control agents like chewing insects, like pathogens or pathogens which are of necrotrophic origin. So some of these chemicals we have used in managing a serious pathogens like Downy mildew in onion and the powdery mildew in roses, because these two serious problems are there in our country. And where I work, there's onion, we all know the name, and it is growing very well in large amount in our different states, like 1,306,000 hectare is covered under this onion cultivation with productivity of 17.17 ton hectare. So the downy mildew that is caused by Peronospora destructor is causing around 12 to 75% of the losses. And severe incidents may include your poor bulb yield, and thereafter the shelf life is very much affected. And the disease actually comes in the low and cool areas. And this warm areas is not required under these conditions. Temperature when it is cool, then circular elliptical lesions, they get rise, and thereafter downy growth, which is underside of that. But when it comes with the humid conditions, it will cover the whole plant, and from the far you can see the blackening of these leaves, infected leaves, yellowing thereafter, and the tip black. This burning we can observe in that. This is the infected plants in the field, and infected bulbs, they show the bottleneck like symptoms. When the infected bulbs, they are infected badly, then there is breakage, girdling from the top end. And this pathogen, which is called as the Peronospora, this belongs to lower fungi group, and is aseptate and high with this haline colorless mycelium. We can see it produces the spongiforms, which bears the sporangia, and it is dichotomously branched. These spongiforms and sporangia are periform or fusiform in nature. And we have studied the pathogenicity test with two different methods: spray and drop inoculation method, wiping off with the this cotton with this distilled water, and thereafter this detached leaf method we have seen under laboratory conditions that detached leaf methods develop more symptoms in early time at 8 days, within 8 days, whereas wiping with the cotton, swabbing with that, and then non-swabbing will develop the symptoms little later. And these are the methods which we have followed because it requires humidity and wool and blotting paper. They have been used with the cotton swab cloth, wetted every time. And these characteristic symptoms we have seen of the downy mildew. And this is in healthy plants. Then we have inoculated. And then the yellowing symptoms, they appear within 11 days. Thereafter, we see the dropping of the these plant infected with the building like these symptoms over there. So we have to compare with these SAR chemicals along with the fungicides. Generally, we are using diethane and some of the non-systemic fungicides, but combi products which were here available with us, we have tried those also, new molecules like Nativo, which is tristrobilin and plus the tibuconazole. And similarly, Cabriotop is the product which is pyrachlorostrobin plus metidum. And Ridomil M gold jo hai, that is giving a very effective result. And we have seen under laboratory condition after 12, 24, and 36 hours, sporangial inhibition was carried out at different these concentrations, and percent inhibition was maximum, only at 500 ppm. These two, Ridomil MZ and then your Nativo and Cabriotop, they have given this some of the sporangial inhibition up to 88.68, but in later and higher doses, 100% inhibition was observed. These are some of the pictures which we can see the desiccation of the sporangia in the best treatment. And similarly, the germ tubing, that production, we have seen that it has reduced in diethane or Ridomil and the Nativo. Or Nativo was not very much effective, but it was also considered to be effective than these non-systemic fungicides. Cabriotop was the next best. And now we have evaluated some of the SAR chemicals in the field. These were tried under the spot conditions also, and the best one we have chosen for these field trials, chitosan, potassium orthophosphate, and the beta aminobutyric acid, which actually trigger the immunity in the plant system and enhances some of the biophysical parameters like phenols, then reducing sugar and all. These are later we have seen in other powdery mildew and rose, this methotrexate system. And we have we have then where is that? Now, these are some of the results we have obtained in this trial. It is going further, so yeah. Now it has come. This you can see the field trial that chitosan, it has given the very good results as compared to your Ridomil and other these chemicals which we have tried. And this was among the chemicals, it was best Ridomil, whereas the chitosan and followed by the potassium orthophosphate, they have very good results under field conditions. Now next is our powdery mildew. This disease, which is very serious and major disease of the roses, which is caused by Podosphaera panosa, is the causal agent. And roses, as we know, they are the #1 flower as the aesthetic value is concerned. And they are also grown in India in many of the places. And now earlier we used to say that Sphaerotheca panosa var rosy was the causal agent, but now on the ITS basis, it has been changed to Podosphaera panosa. And symptoms of the disease is very well prevalent on almost all the plant parts, right from the leaves, flowers, these birds, and thorn, everything is infected by the white powdery growth of the pathogen, and which is actually the conidia. And here also, when we study that your pathogenicity says, then we have seen that within 7 hours, 7 days, the disease is prominent on this. And RAPD was performed by collecting different isolates from different regions. And we have seen that out of 12 these primers used, only 7 that were shown polymorphic this pattern like this, you can see. And we have observed that some of the isolates, they were clustered in the same group, whereas others, they have shown different distinct behavior in that. So here also we have tried some of the fungicides by slide spore germination method, and conadial germination and conadial germination inhibition as well as the germ tube length was measured. And here also 100% this inhibition was observed in case of difenconazole, and it was followed by native or trifloxyauxtovin plus tibuconazole is the next best, and thereafter tibuconazole, because all these chemicals they are new, we have tested. And here we can see the germ tube length, and scheme this hyphae is coming emerging from that, and the initiation of the episodia formation, and thereafter full episodia formation is there, and there is reduction of these. And so whenever we have treated all these conidia under this microscopic examination, we have seen that their germ tube has reduced. And similarly, these chemicals, they have been tested in the field conditions for continuously for 2 years, and we have seen that disease incidence or severity that's very low in case of this difenconazole, 11% only, with maximum this disease control 85%, followed by this nativo or the combination of these two chemicals and the tibuconazole. And these are the field effects. You can see very well this control is full of this powdery mildew, and whereas in others there is clean these surfaces of this rose leaves. And these posts thereafter, when we have sprayed this, and we wanted to know that after this how these chemicals, whether they are antisporulent having those properties or not, because after one first spray we have given, and alternative after 2 days of intervals up to 8 days, because generally it happens that the disease usually appears within the 8 to 10 days interval. So we wanted to know whether these chemicals have the antisporulent activity or not. But we have seen that these also possess those activities, and very less infection have been recorded in difenconazole, and similarly in other similar trend was observed. Now, the SAR chemicals, which we have chosen for that at 2 different doses. Salicylic we wanted to compare, because 85 to 82 to 85% we have seen the reduction in this disease level overall. But in SAR chemicals, we have seen that 78% reduction was observed in the dipotassium orthophosphate, which was best at higher doses, 0.75, as well as lower doses also, around 74% infection. This reduction is seen with the minimum disease level. And salicylic acid was next best, followed by aminobutyric acid. But potassium bicarbonate, usually as a old practice, we have seen that it has given us 66.7% like that in 2017. Similarly, around this much percentage of reduction in the disease level have been observed. So these are the results of dipotassium orthophosphate, similar to that salicylic acid. And some biochemical parameters we have studied, because they are the indicators whether this systemic acquired resistance has been achieved in the plants or not, which are treated with this SAR chemicals. So we have studied some of the reducing sugar content, then non-reducing content also in all these SAR chemicals when we have sprayed them before we have recorded, and thereafter, after 2 days of interval, we have seen that there is a gradual increase in that level of that this reducing sugar content, and thereafter at the bud formation it was reduced, and after bud formation, after 1 month of that, it was drastically reduced later on. It means we require another dose of the foliar application of these chemicals. So non-reducing content was also similar in this, but minimum was recorded in the calcium silicate and highest was recorded in the dipotassium or phosphate, followed by your salicylic acid. And similar trend we have seen with the polyphenol oxidase activity, which was highest recorded in potassium orthophosphate 0.531 and salicylic acid 0.498. Similarly, with aminobutyric acid 0.475. And peroxidase activity in rose leaves after this was also recorded highest. A similar trend have been observed in almost all these are the enzymes which we have been investigated, because this is a phenyl alanine ammonia lyase activity we have observed, and it has also increased up to the 6th day of this first strain, and thereafter at the time of bud formation, because as the reproduction system starts, there is reduction in this resistant chemicals which has been induced in that, because this is only active before the bud formation, and thereafter we have to spray again with this foliar applications. Total phenol content, which is a good parameter, was the resistance in the plants, that is also increased in all these treatments. So in nutshell, and my conclusion, we have seen that 3 protective sprays of the chitosan or potassium or dipotassium orthophosphate at 3 to 4 stage of that plant, if we spray them, then challenge those with this SAR chemicals, and thereafter when there is disease appearance at 15 days interval, we go with that, we can stop the infection, and this way we can reduce the chemical this application every time, because chemicals are associated with various adverse effects on the plant as well as human beings and the environment as a whole. So we want to minimize and include these SAR chemicals in the integrated management program. So similar trend we have seen, but we have seen that high doses, they are more effective in case of salicylic acid and the aminobutyric acid as compared to the dipotassium orthophosphate, where both the concentrations have equal effect, and maximum antisporal activity was also observed in the fungicide, a new chemical which is being now used in the protection of this roses against the powdery mildew pathogen, and all these biochemical parameters, they have increased in case of this dipotassium. So naturally we can use this or recommend it to our farmers that dipotassium orthophosphate or salicylic acid can be used against these controlling of this powdery mildew pathogen. Thank you very much.